my hands up and look at Ray's camera.
Hello and welcome to another session of ASM Live, coming to you a bit late this time from New Orleans during the 2011 General Meeting of the American Society for Microbiology. I'm Jeffrey Fox, the features editor of Microbe, the monthly magazine of ASM, and your host for these sessions. And before we start talking about science, I want to remind those of you who are watching that if you have questions to bring to either of our presenters, use hashtag ASM 2011. During this session, I'll be speaking with Carolyn Harwood of the University of Washington in Seattle, and with any luck, Timothy Donohue of the University of Wisconsin, who also participated in the session and has been detained there, but we hope he'll be joining us. But to begin with, I'm here with Carolyn Harwood, and I would like you to spend a few minutes summarizing your findings, speaking about the highlights and implications.
Okay, thanks, Jeff. Well, I am very interested in biofuel production by microbes. And I'm particularly, I use one type of microbe for this work that can convert the energy in sun to biofuels. And the biofuel that we concentrate on is hydrogen gas, which can be used in conjunction with hydrogen fuel cells in automobiles. To convert the hydrogen to an electric current, and the electric current runs the car, runs the engine, and the combustion product is water. So this is environmentally friendly fuel. It's a possible transportation fuel for the future, and right now we can produce a lot of hydrogen, but it's produced from fossil fuels. And we'd like to find a way to produce hydrogen biologically so that we wouldn't have the problem of dealing with our dwindling fossil fuel supply.
Okay, well that's a good start. Let's talk a bit more microbiology. So tell us a bit about the microorganism you're using, and what some of the, where it stands in terms of being a practical producer.
Being a practical producer, yeah. So there's several different kinds of microbes that can make hydrogen, and the agent of hydrogen production are certain kinds of enzymes. And the one kind of enzyme that we concentrate on is nitrogenase, and there's also another type of enzyme called hydrogenase. Both types of enzymes are very sensitive to oxygen. So the particular kind of bacterium that we work with operates and generates energy from light in the absence of oxygen, and so it doesn't have the problem of protecting these oxygen-sensitive enzymes that make hydrogen. There are other bacteria that use light, cyanobacteria, but they produce oxygen at the same time, so they sort of poison the enzymes. So those guys have certain advantages in that they can get electrons to make hydrogen from water, but the disadvantage that the enzyme that does it gets poisoned are guys which are called purple down sulfur bacteria, operate anaerobically in the absence of oxygen, but they have the disadvantage that the starting material for hydrogen isn't as plentiful as water. But the starting materials that they can use are waste products, industrial waste products, products that are found in municipal waste. So we'd like to think we could convert a waste stream to a useful product with these bacteria. Oh, and here's Tim right now.
So, Tim Donohue, as we
Sorry.
almost promised, is now joining us. He's from the University of Wisconsin, and a couple of minutes ago I invited Carolyn to give a brief overview and summary of the highlights of her work. I'd like you to do that not for Carolyn's work, but for yours. And if you don't mind, take a couple of minutes to give us an overview.
So what we've been focusing on is learning from purple bacteria the logic that cells use to put electrons onto compounds. So that may cause people to go to sleep, but in fact, if you're talking about making just about any biocommodity, whether it's a natural product or a biofuel or a commodity chemical, you've got to set up a superhighway to get the carbon skeletons where you want them, and you've got to deal with whether those compounds have whether those cells have electrons available to reduce them to make anything like ethanol or hydrocarbons or anti-microbials, or one of the most reduced things in the world that people are worried about is hydrogen. So if we don't understand how cells traffic reducing power, we're going to have a problem making any of these types of biocommodities that we would like to in microbes.
All right, so let me follow up. You put that in pretty abstract terms.
Sure.
And in a very general way. Do you have a front runner, a microbe that's particularly adept at doing that and is suitable, or is it a mix of different species that you're interested in?
So
what's your focus?
So to put it in a real life way, and maybe not be as abstract, if you think about Saccharomyces, which makes ethanol in the wine industry or in the beer industry.
I think about it all the time.
And we think about it in Wisconsin quite a bit. Mm-hmm. If you grow them anaerobically, they put their electrons on a compound called pyruvate, and they make ethanol, and we either run a car on that or we enjoy it with dinner, okay?
Right.
If you let a little bit of oxygen into that culture, you get vinegar, okay? So that is the fundamental issue that we're trying to deal with and unravel, is the logic of when they're faced with different places to put their electrons, where will they go? So we are using photosynthetic bacteria in the lab as models to study that. We are also in the lab using photosynthetic bacteria to put them onto hydrogen. Kerry and I have both worked on integrated fuel cell technology where we can oxidize that hydrogen into electricity. And we're starting to move also within the context of the DOE Bioenergy Center that I have the pleasure to run, putting those electrons into hydrocarbons and other fuel molecules that we could burn.
And the important thing that Tim and I have in common is that we're very interested in using the energy from sun to power the process.
Because it's free. It's one of the free energy sources that we have on the planet.
Well, I know, at least, I don't think we tax it even, solar energy is free, but you also have to feed in some chemical skeletons, and that, getting those two, we don't want to say elements, but two components to work together can be part of the big challenge that you're faced with, right? And I'd like to know more. Let's shift to Carolyn for a minute on that, because your organism is not exactly fastidious, but it is a bit choosy, isn't it, in terms of what it will grow on?
Actually, that's one of its strengths. It's not choosy at all.
Oh, okay.
Yeah, it will grow on many, many compounds, different kinds of compounds that are widely present in the environment, including the big one is it grows on aromatic compounds, which are derived from lignin, which is the second most abundant polymer on Earth. So there is a rich source of carbon skeletons available in nature that can be fed into the bug. And then the bug is very, very adept at sort of slicing and dicing the compounds.
All right, and it can deal with the sort of the gemisch that lignin is. It's not a single chemical entity, it's a whole mixture of exotic ingredients.
We
right?
need to lignin's a tough nut to crack, but it needs to be pre-digested with enzymes and things. But then once it's digested down to some of its smaller constituents, then they're very happy
then
with
it runs well.
Yeah.
But one of the things that had me a bit confused when I read through a summary of your work is that you talk about growing cells under starvation conditions. So I'm not quite sure how you get the right balance of producing a lot of hydrogen if you at the same time are starving your cells and asking them to slow down their metabolic activity.
That's an excellent question. And it gets to what Tim was talking about actually, is we talk about moving electrons around because that's how energy is represented. Things that are electron rich or energy rich. And so what the challenge is to feed a compound that's not very valuable into the cell and get cells to rip electrons off it and then move them, reconfigure them somehow to produce an energy-rich compound. And so you want to use cells that that are metabolically active but not growing to do that. Because otherwise, if the cells are growing, they grab up most of the electrons for themselves to increase their biomass. So the idea is we want to just use the bacteria actually as biocatalysts to convert one thing to the other without the cell sucking off anything for its own use.
And so I think Kerry has really hit the nail on the head. What you really want to think about is how do we optimize microbes as biorefineries, as little biorefineries.
Refineries, yeah.
And if they're left to their own devices, sometimes the way they operate in nature is to make siblings and go off and multiply. But in an industrial sense, we would like to control sibling generation and maximize the eating of the food, and instead of and making products. So just like me, if I eat too much, I get fat. We would instead of getting bacteria fat, we would like to think about making them fat on products that society can use. And these photosynthetic bacteria have the advantage of getting a free lunch from light, but also having a voracious appetite of using the sugars that you'll get in plant material or the aromatics that will come from lignin, and also the waste products that we'll get out of other industrial fermentation. So if we're making ethanol in E. coli and yeast, it puts off byproducts, and we know that these organisms will actually eat those byproducts and convert them into a fuel. So you make a second value-added chemical out of the waste products of the first thing. And then the other thing that they do is they have the ability to assimilate CO2 or to sequester CO2. And the hope is that if we can rationally understand all of this and engineer them, we will be able to take the sugars that are cheap, the waste products that are cheap, and the CO2 that comes off from making a bottle of beer, because when you open the cap, it goes poof, right? And take that CO2 and also bring it into the cell and make it into a product to increase the sustainability and the green quotient of the whole process.
All right, well that all sounds like a wonderful, complicated in terms of its organization. I want to digress in a sense. I'm curious to pose this question. In the yeast setting where you're fermenting and producing alcohol, you have a pretty simple, straightforward set of desired products, and you know how to control that. In the situation you're discussing, when do you decide that a cell-free system would be preferable to where you wouldn't have that problem of the cells wanting to replicate, divide, make exotic products that deal with other parts of their life cycle. Is that a feasible undertaking, or is that too far in the future?
You mean not having just having the enzymes? Or
However you want to organize it. I really don't know what the ideal condition short of a living cell would be. But
well, that is the goal of some people, to develop biomimetic materials that sort of mimic photosynthesis and then can transfer the energy of photosynthesis to an inorganic catalyst that could produce hydrogen. And that kind of work's ongoing, and some of it looks like it's going to be very promising. I mean, I think though that those people are taking their design principles from the microbes. So they're taking what they learned from the microbes and trying it to move into a more inorganic biocatalytic setting using the same principles. So that's not what Tim and I work on, but we hope to contribute to the foundational knowledge that helps other people to move in that direction. And whether it will be successful or not, the jury's still out.
okay,
And whether the half-life for getting there is 5 years or 25 years, I think time will tell. But I think Kerry hit the nail on the head again, and that is to say that when we talk to people who have those as goals, and I think they're very good goals, you constantly hear, well, what are all the components that you need? And how do you prevent these critical components from falling apart after 100 days in one of these artificial bioreactors? And that's really at the fundamental research level that we're trying to contribute, getting a list of all the parts, and then knowing how they're assembled and what you would need to keep them stable and active. And can we then translate that into an artificial device? I think time will tell.
all right. Well, in a way, let me go through another door to get at some of the things I'm curious about, and that is, what are the bottlenecks here? Is this sort of getting the right balance between having a cell that's in stationary phase and might, in a sense, be at risk for poisoning itself because it's not growing, or are there other problems that you're faced with in terms of getting good production of hydrogen or whatever other electron-rich product you're after? Are there important bottlenecks here?
Well, yeah. I mean, you can sort of count up what goes in and what comes out. And
what kind of efficiencies are we talking about?
well, we can get pretty high, like 90% of the electrons we feed in, we can get out as hydrogen, for example.
Well,
so
that
it's
sounds
pretty
impressive.
efficient.
Yeah.
But it's kind of like, you know, we get in a car and we drive it around and it works really well and that's fine. But then when it breaks, what do we do? Then you have to go to a mechanic that knows what all the parts are and how to fix them. So a lot of these systems, people already, not our particular system, but people do a lot of industrial microbiology processes and they work. The problem is when they don't work, you don't always know how to fix them unless you know what the parts are and how they're wired together. And so I think we're interested in knowing what the parts are and how they're wired together.
Well, does your system break down sometimes? You've
Oh,
got it
yeah.
up and running and then something goes wrong. What goes wrong typically?
Well, we're making them do something they don't want to do, particularly. So
Why don't they? I thought this is part of their natural cycle.
yeah, but we're forcing them to go to extremes, you know, like, so they don't want to like take all their electrons and put it onto hydrogen because they could use those to better advantage to like sort of maintain themselves. So then you get mutants appearing that can't that stop doing it.
They're kind of like kids. If you try to force them to do things they don't want to do, they will figure out how to take a different path to subvert what you're trying to train them to do. And sometimes knowing as much about where they will go will allow you to improve the process, because you take that choice away from them genetically or metabolically in this process. I've yet to be able to figure out how to do that with my children, but in principle we can do it with bacteria.
All right, well, I like this idea of treating your microorganisms like your family. That's very appealing, but give me, once again, I want to tease out from you name your microbe for one example where the kid didn't behave and you had to send him to boot camp or something. I don't want to go with this metaphor too far. We'll be lost.
Well, we can do it with your kids.
I can give you an example.
Well, but good. Well, I want you both to give me examples. That's your assignment.
All right.
Think of me as your father for a minute. Oh, that got them both. They're dumbfounded. Okay.
Okay, so
think of me as your friend.
Okay, so the enzyme that our bug, Rhodopseudomonas palustris, uses to make hydrogen has another activity, and that activity is to convert nitrogen gas into ammonia. And hydrogen is an unintended consequence of that reaction. So what the cells usually want is they want the ammonia, and the enzyme doesn't work very well, so it always makes hydrogen. But they don't really want to be releasing the hydrogen because that's something that's valuable to them to use in another context. So you have to figure out a way. So whenever you try to get them to make hydrogen, only hydrogen, they won't do it unless you throw them at a bottom of the pool, and unless they put on a scuba tank, they're not going to survive. So you figure out conditions where you incubate the cells, so unless they make hydrogen, they're not going to survive. And so in that way, we select to get them to do something they don't ordinarily want to do. And then when we go in and look to see what happened so that they'd make hydrogen all the time, we can see that they acquired certain mutations. And then we can go in and genetically engineer the bacteria so that we can stabilize those mutations so that they can't decide that they don't want to make hydrogen anymore. So I think that would be an example of what you asked.
Oh, right.
Yeah.
A good example.
And so then another thing that I think both Kerry and I talked about is there are these competing paths that these children can go down. If they have a lot of electrons, they can put them into making a fuel that you want, or they can put them into making some other polymer that needs to get electrons. And in a culture, they may make the fuel for a while, but then something may happen in a wild-type culture, and a genetic switch will be thrown, and they'll make this polymer instead of making hydrogen that has less commercial value in the liquid transportation fuel industry. But you can go in and you can inactivate the genes for making that polymer so that they no longer have the ability to put their electrons on that compound, and you're kind of training them or forcing them to make the compounds that you want.
It's like a parent playing favorites with a kid. They're kids. That's not a good sign. But in microbiology, it works sometimes. Jim, you're signaling me. Do you have a question from the internet? Please share it.
Hold up. Yes, we do have a question from Twitter. A Twitter user, and stop me if forgive me if I get this wrong. Rody Redo wants to know, what kind of breakthrough are you looking for in order to apply the results of your research in a real-life setting?
So I would say not in the hydrogen field, but at least in the work that's going on at Madison as part of this bioenergy center, we have already taken this concept of diverting electrons down the right superhighway into making yeast and bacterial strains that are more efficient at making ethanol from various sugars. I didn't talk about it today in the context of my talk, but we have demonstrated that you can funnel electrons into the appropriate pathways to increase the ability of bacteria and yeast to make fuels using the types of approaches that Kerry and I both talked about today. And those technologies are being patented for future commercialization.
Can I ask, as a follow-up to Kerry, is it possible to siphon off or trap the hydrogen that your cells are producing? Does that improve the efficiency or unpoison the process? Or do they need some of that hydrogen and that's not a good way to go?
That's a great question. No, they don't seem to get inhibited too much by the hydrogen, which is actually a feature of the enzyme that's an advantage. But you can siphon it off and they don't mind.
They don't mind.
And it's easy to separate it out because hydrogen just floats away from everything else.
And there's no comparable way in terms of inputs to keep them from generating nitrogen, or do they still need some of that?
Oh, well, these mutants that we had that put on their scuba tanks,
I'm having a hard time visualizing
to make
that,
hydrogen, so they had to make hydrogen, have to make hydrogen. It turns out that they synthesize the enzyme that makes hydrogen all the time, and we don't let them have any nitrogen gas because we want them to make pure hydrogen, which they will do if you don't give them nitrogen gas. But we can just give them a little bit of like ammonia in their medium and they'll use that as a nitrogen source because these mutants will no longer be inhibited by ammonia. So.
Okay.
But Jeff, I think you put your finger on an interesting issue, and that is that these bacteria, like many bacteria, do have the ability to actually use the hydrogen for energy. So you can set up a futile cycle where you're trying to make this high energy compound and the beasties have the ability to oxidize so you don't get as much. So there's
Does it make them sound funny when they inhale it?
so
That's helium anyway.
That's helium. There's three ways around that. You can either cripple the hydrogen utilization capability, or you can do exactly what you were alluding to, and Kerry mentioned, you can let them off-gas it and capture it as a gas stream. Or Kerry's lab and my lab and others have actually shown that in these cultures you can put anodes and cathodes and put fuel cells, and they will actually oxidize the hydrogen in a fuel cell and generate electricity on site. So in one pot you can take sunlight, waste renewables, and generate current out of it.
Oh, that's pretty exciting. You could have that in your car and
Yeah.
really have a self-contained fermenter-driven automobile. We have a question in the room. Please.
Michael Lange from German Public Radio. I have a question concerning synthetic biology. You sometimes mentioned it. There are people in synthetic biology saying, if the bacteria from nature are not working good enough for us, let's construct our own synthetic bacteria, and they may be much better than bacteria in nature. And you are working with natural bacteria, and you are saying they are doing a good job. What is your opinion? Can synthetic biology in near future lead to better bacteria?
Well, I've been a biologist too long to be arrogant enough to think that we can synthetically produce a bacterium that does the diversity of things that we know bacteria can do, in particular photosynthetic bacteria. So I suppose if you had a very directed function that you wanted a synthetic bacterium to do, then that would be practical. But I think when you get into really complex functions, like photosynthesis, for example, and the metabolic networks that we know have to take place to produce some product, I think that puts a synthetic biology application off much further into the future. That's my opinion.
Yeah, and I agree with that, and I am a microbiologist, and I love the diversity of microbial lifestyles. But I will also say that the last speaker in the session this morning made an equally valid point, and that is that in World War II, society increased the production of antimicrobials over 1 million-fold in order to provide antibiotics to treat soldiers in battle. And that is an example of a specific trait, using Kerry's definition, that I think microbiologists have been able to latch onto and evolve strains to do things for society. And there are many other examples where we can and will be able to replicate that.
Yeah.
It's an issue of taking the problem that you want to improve down to the simplest set of equations and reactions and getting an understanding of it and being able to improve the production of that compound that you want. So I think we will be able to get there. The antibiotic story is I think, a good one. It's a large practical application. It's also a good one because it's an old one and it predates, synthetic biology and genomics. But the tools are now there to go at this in a very different way than we did in the case of penicillin in World War II.
Well, good. I think we've been talking for about as long as we usually talk in these sessions. We've been a bit on an odd schedule. Let me offer you both an opportunity to give a kind of a final thought, take-home message, maybe even give us some perspective in terms of years in the future in which some of these projects are likely to turn practical, just so we're reality-based, and then we'll close down. So, Tim, since you got here late, we'll let you go first.
So I want to come back to I think, an excellent point that Kerry made. Microbes have been on this planet for billions of years, and they have shaped everything that we have around us. They are the master chemist of the universe. And I think, I said it this morning, we are now, I believe, in a renaissance period for microbiology, to be able to take advantage of all the genomic information that we have and look at it as blueprints and see how we can understand microbial life and the implications for how to improve that, how to take those traits and improve the well-being of society is there. And I think from where I sit, some of the very exciting things that we're going to see coming on board very quickly are things like the production of commodity chemicals and fuels that currently come from fossil fuel are going to be out there on the landscape in 5 to 10 years as things that will help allow us to use fossil fuels, the things that it can only be used for, and not have to use it for many things in everyday life. So I see a lot of exciting things on the horizon, taking advantage of these master chemists.
Kerry.
Well, I think Tim said most of it, but I would just add that I've been a microbiologist for a long time, and when I started microbiology, I was really excited. I thought it was just the most interesting thing to study. But I gotta say, I just turned 60, and I'm more excited about it than I've ever been. I just think this is the best time to be a biologist, and a microbiologist in particular. And if I could do anything, it would be to communicate my excitement about how much I love microbiology and think that it is really the science of the future.
Well, I think you've done a pretty good job there of conveying that excitement, so that's great. All right, I've been talking with Carolyn Harwood of the University of Washington, and Timothy Donohue of the University of Wisconsin. That concludes another session of ASM Live. Thanks for joining us.
Thanks a lot, Joe.
Thanks.